Hi guys, we're back looking at this idea of protein synthesis and looking at transcription and translation with the idea that we want to differentiate. We want to make sure we understand the differences in how these processes differ as we look at bacteria versus eukaryotic cells. Okay, so one of the things that we see that is absolutely a difference between bacterial and eukaryotic cells is that the DNA code actually has extraneous information, non-coding information in the DNA when we look at eukaryotes. When we look at prokaryotes like bacteria, they do not have this extraneous non-coding information, right? So just to give an idea of what we're talking about here we have our DNA. And our 5' and 3' ends, we know that that's anti-parallel. And here they're saying that there's a bacterial gene here, okay? And a coding region in blue that must be the code for the transcript, right? The code for the RNA that would be produced from that code. And when RNA is produced, of course, we'd expect for transcription to occur, RNA polymerase to come in and sit down on that code and read it off, right? Down here at the bottom, we have what we would expect with a eukaryotic gene. Again, we see the same basic setup with 5' and 3' at the end, but we see that we have these non-coding regions, and they call them introns. Think of them as junk that's in the way, right? It's in the way. It's introns that are not coding for a darn thing, okay? They're not going to ultimately result in an amino acid at all, right? Because these are non-coding. Versus we have coding regions that we call exons, okay? So these exons are going to be coding for actual parts of that protein. So big difference between the two. We see introns that are in the way, that are extraneous non-coding regions that we find in the eukaryotic DNA, and we find none of that situation, only coding regions within the bacterial DNA. Now there is a lot of thoughts about why it is that we have this kind of junk DNA almost, right? That's in the eukaryotic DNA chromosomes. One thing is that they think that perhaps these were once coding for something, and through evolution they were not as useful, so they weren't ultimately used as part of the gene, okay? We see some proteins that are produced, like antibodies, that will use various portions of the coding regions sometimes, and other portions of the coding regions other times. So this suggests that we could actually see variation and change, perhaps, over time as well. Another little tidbit, a little piece of information, those non-coding regions are actually the regions that we use when we do DNA fingerprinting to like see if, if this guy is the father, right, of someone. They might take a sample from the blood or a mouth swab or something, and they look at the non-coding regions to determine if these people are related. The reason they wouldn't use the coding regions is they'd be too similar, right? These non-coding regions are able to change without a huge impact on the very important coding regions that produce a functional product, right? So they look at, I think it's like 13 locations on those non-coding regions when they do that kind of thing. All right, so again, let's look at another difference between prokaryotes and eukaryotes. We know prokaryotes don't have membranous compartments, and eukaryotes do. We know that the DNA is in this nucleoid region in a prokaryote, but there's a separate membranous compartment within a eukaryote for the DNA. So transcription, by definition, would have to occur within the cytoplasm of the prokaryotic cell or bacterium, and would have to occur in the nucleus of a eukaryotic cell. So there's one huge difference where the transcription occurs. Now, another huge difference would be the fact that there is non-coding information in a eukaryotic cell gene. And that non-coding information would be still in an RNA transcript when that RNA transcript is produced through transcription. So what we see with a eukaryote is a need to remove that extraneous material, to splice out all of those non-coding regions that would be in the RNA. Okay? So with a eukaryote, RNA processing is necessary where we would splice out the non-coding regions from what we call a pre-mRNA molecule, an RNA molecule that has meaningless code in it. Okay? So that's another big difference. Finally, a huge difference that we'd see between them is that in order for translation to occur in a eukaryote, we need to bring that RNA transcript out of the nucleus to have translation occur, because the ribosomes are found in the cytoplasm. Okay? However, we don't need to worry about this in a prokaryote. A prokaryote, the DNA is already in the cytoplasm. Transcription and translation can occur simultaneously. Okay, so as a transcript is being produced, an mRNA transcript, the transcript can be used by ribosomes that latch on and start reading. Okay, so we can have a very quick and efficient production of a transcript and translation to make the protein product. Okay. So here we have an image that is showing the mRNA molecule. So this is mRNA here kind of strung across. And it looks like a bunch of beads on a string, right? Because what has happened here is ribosomes, so this is German rendition here, but the ribosomes are going to latch on all the way up and down this mRNA molecule. And then you can see here that polypeptides, these proteins, are going to snake away from those ribosomes and produce the very same protein from this mRNA, right? Because the mRNA is coding for one thing. So here, it's making lots of proteins. Now down below, you can see what's more is as transcription occurs, the RNA polymerase is here. So it's reading off this transcript, this mRNA, and as that mRNA comes off of the DNA strand, we see that the ribosomes have attached, right? So again, we have this idea where we can have simultaneous transcription and translation within a prokaryote. All right, another major difference between bacterial cells and eukaryotic cells is that not only that there is no non-coding information, it's all coding information, right? But also that several genes are often packaged together in one location of a prokaryotic genome. It's very efficient with its placement of genes. If genes have all a function that have to do with the same basic process, they're oftentimes placed end to end within the chromosome. Okay, and we call that an operon. Okay, so an operon is going to have several genes, like these genes here, A through E. And let's imagine they all are related in that they all play a role in producing enzymes for lactic acid fermentation, right? We know that fermentation involves more than just one enzyme, right? So several enzymes would be produced. Maybe some other enzymes that allow the cell to get lactose into the cell, okay? But everything to do with the lactic acid fermentation. Well, they could be linked together like you see here. Okay, so all of those coding regions would be linked together, and it would ultimately produce the products that would be useful when lactic acid fermentation is going to take place. Okay, so let's call that quits, guys, and we'll talk more about the genetics going on in here and how it is that operons actually turn themselves on and off next time. All right.